Okay, let's make a start. First of all, thank you guys for coming today. I know it's a really, really hot day outside, but I do appreciate you guys taking time out of the day to come and watch what is now our second taster medicine lecture. Okay, so the point of these taster medicine lectures is to kind of give you guys a feel of what you'll be learning as future doctors, future medical students, or even future biology, other science degree students, including biomed, biochem, those kind of things. So it should be interesting for you guys. So a bit about me to start with. My name's Ephraim. I'm a medical student studying at Barcelona London School of Medicine and Dentistry. I've been tutoring for about 3 years now, and throughout the summer I've been dedicating my time to making sure students like you make sure they get the best predicted grades in their September mocks or even next year in their real A-levels. So I personally achieved 13 A-stars at GCSE and then went on to achieve 2 A-stars and an A and A-level, getting 3 out of 4 at my first medical school. So a couple of house rules before we start. Obviously, please stay muted at all times, switch off your video so there's no interruptions during the webinar. If you guys have any questions, this will be completely interactive. Definitely send them on the chat during the lecture or towards the end, and we'll answer them throughout as we go. The slides will be available at the end of the lecture, as well as the recording of this lecture on YouTube. So what you're about to learn is kind of year 1, year 2 level of medicine content. So you're not going to understand all of it, but it will stay relevant as much as possible. So I know a lot of you are doing A-levels at the moment, so I'm going to keep this as relevant as I can to you, so that you can go and answer those kind of application style, suggest style questions that do come up a lot in your A-levels. So obviously I'm not a lecturer in medicine, so anything I do say in this webinar does not substitute for any proper medical advice. Okay, let's start. You're going to be a scientist or a doctor or even a dentist in a couple of years' time. And you're going to be battling with loads of diseases. And probably one of the most common diseases is disease caused by a pathogen, where that could be a virus, a bacteria, or whatever it may be. You're going to be battling with a lot of pathogens. And the challenges of battling with pathogens is, first of all, there are so many of them. There's millions of pathogens on Earth. And we need to identify all of them, some that have not even existed yet, and also be able to treat them in case they do infect humans. And we're all pretty much okay with this at the moment because we're all currently in the hold of coronavirus at the moment. So this is the first of this 2-part webinar series. So this will be about bacteria, how you tackle bacteria, kind of antibiotics, those kind of things. Our second webinar will be on viruses, which is all really interesting stuff. Let's start with bacteria then. So antibiotics are probably one of the most common drugs you'll be prescribing as doctors or as scientists you'll be investigating. They're really, really common. And the challenges with dealing with bacterial infections is there are so many species of these bacteria. And you need to be able to identify which species of bacteria the person or your patient is being infected with, and also give them the right medication, in this case, antibiotics, to kind of overcome bacterial infection. And this all really much relies on having a good, sound biological knowledge around the structure of these bacterial cells. So let's start with bacteria. Let's talk about its general structure. And you guys should all be aware of this for your A-levels. So this is a really generic structure of a bacteria. You have an outer cell wall. This cell wall is not made of things that eukaryotes are made of, so cellulose or chitin. It's made of things that only prokaryotes, such as bacteria, are made with. So in general, this is called a peptidoglycan or a murin cell wall. You then have an inner cell wall, cell membrane, and then you have some genetic material, so circular genetic material, the definition of prokaryotes. You also have these small circular rings of DNA called plasmids. And they're all surrounded in cytoplasm, obviously. We also have small 70S ribosomes. So again, in A-level, you learn what 70S or 80S means. In A-level, you should be aware that prokaryotes, including bacteria, are 70S ribosomes. And humans, plants, they all have 80S ribosomes. This is going to be really important, so hang on to that fact. We also have a flagella, and also a whole host of enzymes working to keep this bacterial cell alive. And this somewhat simple bacterial cell can cause so much harm. And there's so many different types of disease that we need to know about. But this is a general structure of bacteria. Let's classify these bacteria, because what you need to do as a scientist or as a doctor is be able to classify every one of these hundreds and hundreds of species of bacteria, identify which one your patient is or your person suffering with, and then give them the correct drugs. And there's so many to kind of choose from and know about. So let's see how, as a scientist, you'll be identifying bacteria. The first, most common classification of a bacteria is this bacteria gram positive or gram negative? And you guys are on Edexcel. You guys know what gram-positive and gram-negative are. But the other load of you that don't know what gram-positive, gram-negative are, let's understand what's going on. So we talked about the bacterial cell wall, the outer structure of that bacteria. And each different bacteria has a different structured cell wall. But we can put all bacteria into either two groups. We can put them into either gram-positive bacteria, so a kind of bacteria with a special type of cell wall, and also gram-negative, which is another group of cell wall structures. So all bacteria fall into one of these two classes, gram-positive, gram-negative. What does that actually mean? So let's talk about gram-positive bacteria. Gram-positive. So gram-positive bacteria have a special structure. And this structure is their cell wall, the outer cell wall is made of two layers, two really thick layers. And these two thick layers are called peptidoglycan layers. They're two thick, really layers, and they're all joined together using peptide bonds. You guys already know what peptide bonds are. I'm hoping the majority of you do. So it's a thick outer peptidoglycan wall held together by peptide bonds. You then have an inner cell membrane just like before. That's gram-positive. So kind of half the bacterial species have this structure. The other half of all bacterial species have a different type of structure cell wall. This is called a gram-negative cell wall. And the structure slightly varies. So your outer cell wall is this time not a thick outer two-layered peptidoglycan cell wall. It's just a thin layer of outer lipopolysaccharide cell wall. This is lipopolysaccharide. And as the name suggests, it contains some lipids. That's going to be really significant in just a while. But just be aware the outer cell wall is kind of made of a thin layer of lipopolysaccharide. And then also, just attached onto that directly, a really thin phospholipid bilayer. However, in that second, we have another second layer of this cell wall. And this time, it's just a thin layer of peptidoglycan. So see how they're kind of opposing. So in our gram-positive, we have a thick two-layered peptidoglycan cell wall with adjacent peptide bonds holding it all together, giving the bacteria structural integrity. And then we have an inner cell membrane, phospholipid bilayer. Gram-negative bacteria, the complete opposite. We have a thin outer lipopolysaccharide cell wall attached with an outer membrane, outer phospholipid bilayer membrane, and then also an inner cell membrane. So the outer component of the gram-positive is a polysaccharide, a really thick layer of polysaccharide. All right, thick polysaccharide. And if you're gram-negative, you have a thin lipopolysaccharide and a thin cell membrane just directly attached to them. So those are kind of the phospholipids. But remember, it's made of lipids, which is going to be really important. And knowing the structure of these cell walls is going to be really important and imperative to knowing, first of all, what type of bacteria your patient's been infected with, and secondly, what kind of antibiotics you give, because different antibiotics react differently, and that some antibiotics will be allowed to get into the cell wall, some won't, and we'll talk about that in just a minute. So that's the two structures you should be aware of for all bacteria. So gram-positive, gram-negative. And what we do with these two species of bacteria, we take a blood sample of the patient suffering with a bacterial infection. We then carry out this process called gram staining, which is the process of identifying, is it a gram-positive or is it a gram-negative cell wall? And there's 4 steps to doing a gram stain. The majority of you in your degrees will do this probably on your first couple months as an undergraduate, but people on Edexcel will already know what this is. So let's talk about what's going on here. So the first step in gram staining is adding a substance called crystal violet, so kind of a violet stain. Okay? And what this does is to both species of bacteria, either a gram-positive or a gram-negative, you both get stains purple. So the outer peptidoglycan cell wall, the outer lipopolysaccharide cell wall, they both get stained purple. So let's draw what's going on at this stage. So if you're a gram-positive and a gram-negative, let's see what's going on. So your outer peptidoglycan will go kind of a bluey purple color. And also your gram-negative lipopolysaccharide will also go to kind of bluey purpley color. The second step is adding a component called iodine. All iodine does is making sure that the stain is soluble, stays less soluble so it can stick better onto that either peptidoglycan or lipopolysaccharide cell wall. So in the second step, both your bacteria, gram-positive and gram-negative bacteria, will both appear bluey or kind of purpley color. The third step, though, kind of distinguishes the different species. So I told you gram-negative bacteria, I told you they have an outer lipopolysaccharide cell wall, which means it contains lipids. And for you guys that know A-level, or just any of you, you know that alcohol, being a solvent, can dissolve lipids. So that means the outer lipopolysaccharide cell wall made of lipids is going to get dissolved in only gram-negative bacteria when you add alcohol. Because remember, gram-positive, they have a thick peptidoglycan, so fully polysaccharide. So at this third step, let's see what happens. Your gram-positive and then your gram-negative bacteria. Your gram-positive bacteria will still remain purple because nothing's going on to them. The alcohol has done nothing to kind of break it apart. However, your gram-negative bacteria, their outer cell wall is going to be destroyed. And all they're going to be left with is an inner cell membrane, or that kind of, a really inner cell membrane. That's all they've got left. The outer cell wall has been taken off because it's made of lipopolysaccharides. And this is really useful. What we do next is add something called a counter stain. So another stain on top of this. And what this does is it only binds onto cell membranes. That's all this safranin component does. It only binds to cell membranes. And we know that gram-positive bacteria, its cell membranes aren't exposed. So it's going to remain purple or blue. However, gram-negative bacteria, we saw that its outer cell wall was broken away and its cell membrane was exposed. We did that using alcohol. So what we're going to do now is add this counter stain, the safranin counter stain. It's a component called safranin. And it has a kind of pinky, reddy color. And what it does is it only attaches selectively onto cell membranes. So what happens is your gram-negative bacteria go red or pink. And now you can see you've distinguished between either gram-positive or gram-negative bacteria. And this is a really powerful tool because certain antibiotics only work for gram-positive, certain only work for gram-negative bacteria. This is a really powerful tool, what we've done. So at this stage, what you've done is identify gram-positive and gram-negative bacteria. So gram-positive are this kind of purpley blue color, your gram-negative are your pinky red color. Someone's asking a question on the chat. Why do you need safranin when you can tell the difference after the alcohol stage? So after the alcohol stage, what you're doing is exposing the cell membrane. The cell membrane is pretty much transparent. So you want to add another stain on top of that so we can selectively increase the contrast of the cell membrane. And that's why gram-negative, being the one that's got an exposed cell membrane, will turn red or pink. So these kind of similar themes of staining appear in A-level biology. So in your first chapter, you learn about microscopy and staining. The same things are going on here. This is just a special type of stain for bacteria called gram staining. I hope that answers your question. So now what we've done is kind of do a 50-50. We know what the overall species of bacteria is. Is it gram-positive or gram-negative? Well, I told you there's hundreds and hundreds of species of bacteria. So we need to be a bit more detailed. We need to kind of classify and pinpoint the exact bacteria. And there are several other types of tests you can do, which are very complicated, so I'm not going to go over it at this level, but eventually you will learn about them. So there's 4 tests: the catalase, coagulase, hemolytic, and Lancefield test. We'll then kind of go and pinpoint which species of bacteria you're dealing with. And that means you can fully identify what the person's symptoms are going to be, and also what antibiotics and treatment you can give them. Another way you can kind of speed up this whole classification process, because let's say, for example, you've got a blood stain, you're a scientist or a doctor, to take this blood stain, which potentially may contain bacteria, and identify what species it is, it's going to take a long time. And we don't have that much time if we're talking about kind of life-threatening bacteria. So what you can do is kind of speed up classification by just doing a simple microscopy test and seeing what the shape of the bacteria is. And it's another way you can classify bacteria. So you can actually classify shape into 3 different types of bacteria. You've got kind of spherical bacteria, known as cocci, in Latin, cocci means spheres. Rods, you've got bacilli, and you've got spiral bacteria. And they all have unique shapes. And different bacterial species have different shapes. They take on different shapes. So you guys have probably heard of Staphylococcus aureus, or MRSA. So Staphylococci, cocci meaning spheres, have a very spherical shape. So we can kind of take almost a guess at what kind of species we're dealing with, and we can kind of rule out all these other bacteria. You guys have probably heard about cholera bacteria. Cholera bacteria has a spiral shape with a tail. We know the person could be infected with cholera. Salmonella takes on this kind of bacilli or rod kind of shape. And what we're going to look at a lot today is something called Helicobacter pylori, H.pylori, a really, really common bacterial infection in the UK. And it takes on this distinguishing spiral structure. So you can see over here, if you were to get this under an electron microscope, you can see that you can pretty much see this, the kind of flagellum of the bacteria. And it kind of resembles cholera. So we can identify this person has cholera. Okay, let's talk about why bacteria are actually harmful in the first place. They're harmful for several reasons. And it depends on what kind of species of bacteria you are. But in general, bacteria can cause damage in two ways. So first of all, let me draw the structure of a general bacteria. So this can be either gram-positive or gram-negative. You can see in this diagram over here, these kind of strands coming out of each of those types of cell walls. Those strands are kind of toxic components. Okay, they're really toxic components. And when they interact with your host cells, the host in your own body, they start to cause damage and apoptosis or programmed cell death. So some bacteria can do that. And that mechanism of using the outer cell wall structure as a toxin or as a kind of harmful thing itself, just by attachment, they're called endotoxins. Okay, so endotoxins, so endo kind of means in or within. Endotoxins work by just attachment. So attachment of the kind of cell wall structures. Since they're so foreign in the body and our body doesn't recognize them, they treat it as a harmful toxin. So we call them endotoxins. The second class of toxins a bacteria can use to create impact or damage inside the body to host cells is actually release kind of protein toxins from their cell wall. So they make it inside and they start to release a whole lot of different types of toxins that are harmful to cells. These can damage white blood cells, they can damage pretty much all your other cells in the surrounding area. So the bacteria releases toxins from within themselves. These are called exotoxins, exo meaning out or not within. So exotoxins, certain bacterial species can release them out, whereas endotoxins, certain other species, just by attachment of their different varied components of the cell wall can cause immediate damage and also apoptosis to the cell walls, to this host cells. Sorry. And when you feel symptoms, for example, of a bacterial infection, you're basically feeling the effects of these toxins. So for example, the apoptosis, the death of your cells, you're going to feel that as these kind of feverish symptoms or other kind of redness, swelling, those kind of symptoms that you feel with bacterial infections. Toxins kind of damaging white blood cells and damaging surrounding areas and your cells, you're going to feel that as well by symptoms. And there's loads of other symptoms that are kind of specific for each bacterial species. Okay. But there's another way you can kind of also further speed up the process of classifying what bacteria you're dealing with, which is really simple. Just identify, first of all, what part of the body the bacteria is infecting, and secondly, what symptoms the patient's presenting with. And they're going to be pretty much different for each specific species of bacteria or kind of groups of bacteria. So let's say, for example, someone's got a skin rash or kind of swelling or pus developing on their skin. You know, it's going to be kind of a skin-based bacteria. And from our previous knowledge, we can try and identify and narrow down what kind of species of bacteria we're dealing with. But let's say, for example, this bacteria is causing kind of symptoms within your stomach. So indigestion, heartburn, those kind of things. We can probably narrow it down to these bacterial species living inside your stomach or some kind of part of your digestive system and causing impact there. And we already know a lot of different types of bacterial species that do that. And that also kind of quickens the process of diagnosing a patient. Because in medicine, in science, with dealing with pathogens, time is kind of life as such. If you delay it too long, this person could potentially die or kind of suffer with severe symptoms or kind of lifelong impacts of these bacterial infections, which is really crucial to understand. So that's why it's really crucial to identify what species the bacteria is very quickly. And that's going to be your job as future scientists and doctors. So let's do a patient case, because I think it's really interesting to see what you do as a doctor or a scientist if you're put in this situation, based on all the knowledge we've kind of built up so far. Okay, so you're going to be a GP. Let's say you're a GP or a microbiologist or something like that. And a patient comes into you, a 23-year-old female patient, presents to, let's say, the GP practice in this case, with frequent loud burping, kind of heartburn-like pain, stomach pain, indigestion, those kind of things. So that's the symptoms she will come to you and tell you. And as a clinician, as a kind of scientist, you'll be aware of kind of common bacterial infections. Okay, and these are kind of really, really classic symptoms of a very specific bacteria that's very prevalent in the UK. And this bacterial species is called Helicobacter pylori, which is the one I kind of told you to remember at the beginning. So Helicobacter pylori, H.pylori, is probably one of the most common bacterial infections around the world. And believe it or not, about 50% of the UK population already has H.pylori in their stomach. And for the majority of people, it doesn't cause problems, doesn't cause symptoms. But for certain people, it can cause very aggravated symptoms. And these, for example, this patient is one of them. So it's a very common bacterial infection, and you'll be used to its symptoms when you're a a healthcare professional. What Helicobacter pylori does is it affects the stomach and the GI tract, the GI meaning gastrointestinal or kind of digestive system. Common symptoms, you've got frequent loud burping or belching. You've got a symptom called gastroesophageal reflux disease, which is basically acid in your stomach coming up your esophagus, and then you can start feeling it in your throat, so kind of a heartburn kind of pain. You also have very severe bloating in your stomach and your intestines. You start to lose weight because these bacteria are going to start eating off your food. And also you could start to present with blood in your stools. So these are all kind of really common symptoms of a bacterial infection called H. pylori, a really common bacterial infection. And left untreated, it can develop into several further really serious side effects and long-term impacts, one being actually stomach cancer. So it's really important to diagnose and correct this. So let's see what's going on with H. pylori. So the word we use to describe how this bacteria is working, what it's doing in your body, we use this word called pathophysiology. So the pathology of the illness and the physiology of your body, combining pathophysiology of this bacterial infection. So as I said before, H. pylori is a really common bacterial infection. About 50 to 60% of the UK population has H. pylori already in their stomach. And about 90% of cases, it doesn't cause any symptoms, but in certain people, it can become aggravated. So you can get H. pylori by kind of bodily fluids, so kissing, saliva, vomit, fecal matter, even contaminated food can all transmit H. pylori. So it's kind of ingestion is the route it takes to get inside. And what these bacteria do, so let's say this is the inner surface of your stomach. So this is the kind of inner surface of your stomach that's exposed to food, and this is kind of the stomach lining. So what I've drawn is kind of zoomed into that area over there. What does H. pylori bacteria does once inside your stomach? So first of all, using its tail, it's going to drive itself and kind of burrow in to the lining of your stomach. That's the first thing it's going to do. And it does this by kind of burrowing a hole through your stomach. And the exact mechanism it uses is by what we talked about, so endotoxins. So remember, endotoxins are kind of the outer cell wall structure that's actually harmful to human cells. And just by attachment, it can start to cause apoptosis of your stomach cells. And what it does is it kind of creates a hole for itself to get in. Okay? So that's the first thing H. pylori is going to do. The second thing H. pylori is going to do is release loads of enzymes. And one of probably the most notable enzyme that's going to release is something called urease. So remember, all enzymes end in ASE. Urease, by definition, it's an enzyme breaking down urea. Okay, urea. And what the function of this urease enzyme is to make the conditions in the stomach more favorable to the survival of this bacteria. You're probably aware that inside your stomach, you've got kind of hydrochloric acid at really, really low pHs. And it's very harmful to the vast majority of pathogens, including H. pylori. So what H. pylori is going to do is once inside the stomach, it's going to switch off, first of all, kind of switch off the cells making acid, and also neutralize all the acid in your stomach so that the conditions are more favorable to its survival. And the way it does that is, first of all, secreting this enzyme called urease. What urease does, so let's see what urease is going to do. It's going to take urea inside your stomach that's kind of naturally found in your stomach and convert it. So using this enzyme called urease, it's going to convert it into two components, CO2 plus NH3. So CO2, the kind of vast production of CO2 causes the burps. Okay, that's one of the symptoms we talked about. NH3, you guys know that NH3 is a base. Okay, that base is going to react with hydrochloric acid and cause neutralization, which means the hydrochloric acid in your stomach, which is kind of acting to digest your food, protect you against infection, is now being switched off or neutralized so that this bacterial species can better survive. Okay. And what happens is your body knows what's going on. It kind of has an idea that the acid levels in your stomach, which can be detected by kind of certain cells in the lining of your stomach, they can detect that the pH in your stomach is starting to rise and the levels of hydrochloric acid are starting to fall. So what it does is it switches on or kind of overreacts all the cells in your stomach and tell them to keep producing loads and loads of acid to kind of combat this, this loss of acid. And when more acid is secreted into your stomach, so too much acid, it causes heartburn, or that kind of symptom I talked about, which is called gastroesophageal reflux, which is basically when acid goes up your esophagus, you feel heartburn. And it does this continuously, and to the point where you get too much acid in your stomach, which can be very harmful. We've got a question in the chat. It says, what does apoptosis mean? So apoptosis is defined as programmed cell death, or where the cells can take a conscious decision to get rid of themselves or kill themselves, to kind of create, to minimize the harm elsewhere. Okay, that's what apoptosis means. And it's not a good thing. Okay, so I hope that all makes sense. We know what's going on with H. pylori. We know what it's doing, what it's secreting, how it's kind of utilizing the environment in our stomach. Since bacteria is here, it needs kind of a supply of nutrients. That's going to take it from your food. So every food that you kind of put in your stomach, it will start to digest it and use it for its own survival. So it's kind of taking over your stomach, which accounts for several of the symptoms we talked about. So gastroesophageal reflux, this is a heartburn, weight loss because you're kind of losing the food here, several other kind of pain-related things in your abdominal area. Okay, now that we know everything that's going on, let's talk about how to treat bacterial infections. And as I said, there's hundreds and hundreds of species of bacteria, all having different structures. We talked about one way to classify all bacteria into kind of 50-50. So let's talk about antibiotics and their kind of general mechanism. There's different classes of antibiotics, and they all use different mechanisms to kill a bacteria. But we can group it into, so all antibiotics can be grouped into two ways that they act. They can either be called bactericidal, let's write this down, so cidal, or bactericidal, or bacteriostatic. And people in A-level, you guys are going to learn about this eventually. But let's see what these two words mean. So remember, all antibiotics can be grouped into two classes, either bactericidal or bacteriostatic antibiotic. So static means stasis or kind of keeping still. So what bacteriostatic does is it prevents binary fission, or kind of the reproduction of these bacterial species. So it slows down reproduction rates. If we slow down reproduction rates, less bacteria are produced by binary fission, you don't feel the impacts as much. That's what some antibiotics do. Other antibiotics work by a mechanism called bactericidal. Cidal, or kind of, cidal means to kill off. So bactericidal antibiotics work by actually physically killing each and every bacterial cell. So it kills. And all antibiotics can be placed into two of these categories. So we know the specific structure of the bacteria. We talked about its outer cell wall. It can be different if it's gram-positive, gram-negative. We talked about the inner cell membrane, what's in the cytoplasm, the genetic material, the plasmids, the ribosomes, the 70S ribosomes. And we can target each of these. Because in a human cell, we don't have cell walls. We don't have 70S ribosomes. And we don't have certain enzymes that only these bacterial species affect. That means we won't be affected by antibiotics. There's different classes. Let's talk about a couple important ones. So the most common, or the first ever antibiotic discovered, was penicillin. And what penicillin does, we're going to talk about it in just a minute, but penicillin works to destroy the cell wall on all bacteria, gram-positive and gram-negative bacteria, with a preference towards gram-positive. And we'll see why in just a minute. So penicillin works to break off the cell wall and destroy the cell wall. And there's also other kinds of penicillin-like drugs. So, for example, amoxicillin, piperacillin, so anything ending in C-I-L-L-I-N-S, or cillins, they all act in the same way, which is to break the cell wall down. We know that ribosomes have a 70S subunit, or they're kind of made of 70S ribosomes. And since humans only have 80S, we can target the 70S and break down the 70S. If we break down the 70S, this bacteria can't do protein synthesis. If it can't do protein synthesis, it can't go through binary fission. If it can't go through binary fission, it won't reproduce. If it doesn't reproduce, you'll start to lessen the impact inside your body. That's one type of antibiotic mechanism. You can also disrupt its DNA synthesis or semi-conservative replication. We can damage that as well. We can stop it synthesizing more DNA in binary fission, which means all the bacterial cells don't have any more DNA. And there's several other mechanisms, but the ones I told you, so destroying the cell wall being one of them, destroying the ribosomes another one, and destroying their DNA is kind of three main mechanisms. Okay. That's three of our kind of classes of antibiotics. Let's zoom into penicillin. This is a really common one of the first ones discovered. So knowing which antibiotic to give, let me just quickly say, kind of relies on cell biology. So you need to know the structure of bacteria to be able to identify what kind of bacteria is infecting the patient. That's why cell biology is really important. That's why, obviously, if you're in A-level, don't dismiss that topic. Okay. let's talk about penicillin. Penicillin and the associated sister drugs, for example, amoxicillin, cephalosporins, amoxicillin, they all work in the same mechanism because they all have the same structure. Let's talk about the structure. So penicillin, amoxicillin, piperacillin, cephalosporins, they all work because they're the same structure. And the structure resembles these. So these are all the 9 types of penicillin sister drugs. And you can probably notice a common theme in their structure, which is this ring over here. So that ring with a nitrogen group there. So you can see every type of penicillin sister drug has the same central structure, which means they work in the same mechanism. And that ring, it has a special name. It's called a beta-lactam ring, beta-lactams. So penicillin, amoxicillin, piperacillin, cephalosporins, and a couple other, they're called beta-lactam antibiotics, which means they all have the same ring in the center. And that means they all work in the same mechanism. Okay. Let's zoom into the mechanism of how these antibiotics are actually working. And I said they work on both gram-negative and gram-positive, but they're preferentially going to kill off gram-positive bacteria. Let's look at how. So what I'm going to draw is a gram-positive bacteria. The same mechanism applies for gram-negative. But let me draw a gram-positive. So we have the outer thick layer of peptidoglycan. Remember, they're attached together using peptide bonds to keep the structural integrity together. And then you have an inner cell membrane, so your typical phospholipid bilayer. And there's a certain enzyme that works to keep the integrity of your outer peptidoglycan cell wall together. First of all, there are bonds that hold the 2 layers together. And I told you they're peptide bonds. So peptide bonds work to hold the 2 layers of the outer cell wall together. They're peptide bonds. And the enzyme that catalyzes the formation of these bonds, okay, that's a special name. So these enzymes sit in the bacterial cell wall and keep the integrity of these bonds, these peptide bonds, which overall keeps integrity of the cell membrane. Sorry, the cell wall, the peptidoglycan cell wall. These enzymes here are called transpeptidases. And the reason they're called that is trans between 2 layers, peptide bonds, they're creating peptide bonds, and the enzyme, so ASE. So transpeptidase is the enzyme that keeps the structural integrity of both species of bacteria, gram-positive and gram-negative, but more specifically gram-positive. So if you're gram-negative, instead of your outer peptidoglycan, it'll be an inner, but they're both working the same mechanism. This is a gram-positive. Okay, what our antibiotics are going to do? Let's talk about penicillin, probably the most famous example of an antibiotic. What penicillin is going to do is work by the same mechanisms of enzyme inhibition that you guys have learned about, so non-competitive and competitive. What it's going to do in this case is block the active site of these transpeptidase enzymes. So if you give a person penicillin or any of those sister drugs, those beta-lactam antibiotics, what's going to happen is they're going to block the active site of transpeptidase. And this is irreversible. So they're irreversibly blocking the active site. If they do that, guess what happens? The peptide bonds holding the integrity of your cell wall start to break apart because the enzyme's no longer there. If that starts to happen, the cell wall basically starts to float away. And you can see the damage that's caused to the bacteria already. The only layer protecting the inner cell cytoplasm, the genetic material, the only thing holding together the integrity of this bacterial cell is its cell membrane. And the cell membrane is very permeable to water. So the water in your blood, the water in your tissue fluid, is going to just simply enter by osmosis. And since the water potential in your blood, your surrounding tissues, is very high compared to inside the cytoplasm, it's going to go by osmosis. Water is going to flood inside the bacteria because the cell wall is no longer stopping water getting in because we destroyed it. And guess what? It's the same mechanism that you guys will learn by an A level. If water enters the cell membrane, it will eventually cause it to burst. Burst is called lysis. So it causes the bacterial cell to lyse or become dead, basically. And if it bursts, the genetic material floats around, the bacterial cell can no longer cause damage to your cell body, to your cells of the body. So we've just killed this certain bacterium. That's how penicillin works, or and this is the drugs as well. Bacteria, they're kind of clever. They're not as stupid as this. They're not going to let a simple penicillin drug hinder their survival. So remember I told you about certain structures inside of your cytoplasm, inside predominantly most bacteria. I told you about these structures called plasmids. In A-level biology, you know about in cell biology that plasmids, they contain genes for antibiotic resistance. Plasmids, they do antibiotic resistance. What does antibiotic resistance mean? They basically break apart any antibiotics you give, which means they'll no longer respond to antibiotics, they'll no longer be killed. And a lot of bacteria these days are becoming antibiotic resistant. And that's a whole nother topic to talk about, which is kind of covered a lot in immunology and A-level. But what certain bacteria do is they develop genes. And these genes coded for by the plasmids allow the coding of enzymes, special enzymes that sit on the outer surface of the cell wall. Okay. And these enzymes, they break certain things down. These enzymes are called beta-lactamases. So we know that all these penicillin sister drugs, they have a beta-lactam ring, that central 3-carbon ring. They're called beta-lactams. And this enzyme that the bacteria produces to kind of kill off all the penicillin-type drugs is called a beta-lactamase. Well, enzymes, remember, they all end in ASE, so beta-lactamase. And beta-lactamase, what it does is the minute you give penicillin or amoxicillin or any other sister drugs, it goes inside, because it's complementary to the enzyme breaks it apart. It breaks apart penicillin and their sister drugs so that it can no longer damage transpeptide by inhibiting it. So that's really clever. But humans are probably more clever than the bacteria. What we've done is come up with an inhibitor drug of the new enzymes that the bacteria has produced for antibiotic resistance. So what we do in modern clinical practice today, which is all kind of done by the work of really good scientists around the world, was to create inhibitors against these new enzymes called beta-lactamases. And these inhibitors, they have several types of names, but one of the most common one, I'm going to write over here, is called clavulanic acid. Clavulanic acid. Clavulanic acid is specific and complementary to, guess what, beta-lactamases. So it blocks beta-lactamases and prevents them working. If it prevents them working, you can safely give penicillin or any of those other drugs like amoxicillin, and it will go and bind onto transpeptidase, inhibit them, break the peptide bonds apart, break the cell wall apart, allow water to enter by osmosis, and cause the bacterial cell to burst. This is all really the fundamentals of enzyme inhibition is working here. That's why A-level biology is really crucial for your kind of further degrees. So I mentioned 2 drugs. I mentioned the first one, the beta-lactam antibiotics, so your penicillin, amoxicillin, and also the enzyme inhibitor for these new enzymes that the bacteria produces against antibiotic resistance, which is called clavulanic acid. So what we typically do in medical practice nowadays is we no longer just give penicillin, or we don't just give amoxicillin in most cases. We actually give both, the enzyme inhibitor first, and then the penicillin, or both together. So we used to give just amoxicillin. And sometimes we do still give it if the bacteria is not a very developed bacteria. But nowadays we give something called co-amoxiclav. Co-amoxiclav means means together, amoxicillin, the antibiotic, and clavulanic acid, the inhibitor of the beta-lactamase enzyme. So we just give this whole drug, co-amoxiclav, which is the inhibitor and the actual antibiotic, amoxicillin, which is a sister drug of penicillin. So it's all to do with simple A-level biology concepts. It's really interesting stuff. Let's go back to our patient. The patient, a 23-year-old female with H. pylori. To diagnose a bacterial infection, I told you can kind of specify the symptoms, the location to quicken the process. And since the GP's probably seen this a lot, and again, as scientists and future healthcare professionals, you'll see H. pylori a lot because it's very common. Let's talk about its structure really quickly. It has a gram-negative cell wall. I told you it produces the enzyme urease to neutralize stomach acid. It has a helical structure, we know about this already, and it has a flagellum. So this is kind of a scanning electron micrograph, you can see the flagellum. So now that we know its structure, we can tailor antibiotics against this now. Okay. So let's first of all talk about how we can diagnose H. pylori. And we can do this by basically the symptoms, and the symptoms relate to the structure of H. pylori. I told you H. pylori makes a special enzyme called urease. Remember, urease breaks down urea in your stomach into ammonia and carbon dioxide. The ammonia neutralizes hydrochloric acid in your stomach, gives the bacteria a favorable environment. So we can test for the presence of urease by simply giving a person urea. We tell them to drink urea. And on that urea, there's a special isotope attached onto it, a kind of carbon dioxide isotope. And what we do is we tell the person to drink this solution. If urease is present, it will break down urea that you've given into ammonia and carbon dioxide. And then the carbon dioxide that you breathe out is a special isotope. We just detect for the presence of carbon dioxide using a simple lime water test. And if we know that urea, if that urea that we've given is being broken down by some urease enzyme, it must mean that H. pylori is present because only H. pylori releases urease. So we're kind of working backwards to diagnose H. pylori. So this is called a urease breath test. We can also take a biopsy, taking a sample of your stomach in kind of more serious cases. We can also test for blood antibodies. So if our immune system is kind of making antibodies against H. pylori, we can detect for those antibodies. We can also do something called a stool antigen test, which is basically testing for small fragments of H. pylori broken down by neutrophils. We can test for that in your stools. If it's present, it means H. pylori is also present. Okay. And we can do that using, again, A-level concepts. So the ELISA test, enzyme-linked immunosorbent assay. So what we're doing is, let's say we're doing a blood antibody test. We're testing for antibodies present or even antigens present in your stool or your blood sample. And this is the ELISA test, and it uses monoclonal antibodies. Let's quickly go through this because I know a lot of you are doing A-level. So what you do is get a Petri dish, you get a sample of their blood plasma or their stools. Let's say we're going to test for, let's say the enzyme, if our immune system is making enzymes specific against H. pylori. So what we do is we can make these monoclonal antibodies that are specific against H. pylori. So what we do is we put these monoclonal antibodies down in our Petri dish. Let's say we're going to test for the antibody. Let's do that. So we're testing for the antibodies being present of H. pylori. So these over here are fragments of H. pylori antigen, and we're testing for the antibodies present in the blood. That means this person's probably being attacked by H. pylori. So we put some H. pylori antigens as basically like a trap in our Petri dish. We then pour some blood plasma. If their blood plasma contains a complementary antibody against these H. pylori antigens, it will selectively bind. So let's say this person's blood plasma contains antibodies against H. pylori. They will bind here. This is known as your primary antibody. So this is a kind of an indirect ELISA test. We then wash this all off to avoid kind of false negative, false positive tests. We then put a solution of another monoclonal antibody, which in this case is complementary to this antigen antibody complex. This is called our secondary antibody. And this secondary antibody has enzymes linked onto its surface, hence enzyme links or the ELISA test. And what this enzyme does is it breaks down a dye solution. So we're going to pour some kind of colorless dye solution. And inside that colorless dye solution, we have molecules that, if broken down by these enzymes, which if they're present, will turn the solution red. So let's say it turns red. We know that there must be some kind of H. pylori specific antibodies present in the person's blood, which means they have H. pylori somewhere in their stomach. That's the ELISA test. That's the kind of mechanism behind the ELISA test. It's more complicated, and we'll learn about the full details if you do A-level biology. But it kind of revolves around major A-level concepts, so monoclonal antibodies, ELISA tests, immunology, those kind of things. This is also, so the ELISA test, might as well just tell you now, is useful in many common things at the moment. So for example, pregnancy tests work by the ELISA test. Urine tests work by the ELISA test, kind of like drug sampling, those kind of things. For pathogens, diagnosing pathogens, very, very much common today. So coronavirus testing. So when they stick the swab in the back of your throat, they're going to do the ELISA test straight up to test for the presence of antibodies or antigens against coronavirus. So it's really important, the ELISA test. Let's talk about the treatment for H. pylori. So we know everything we need to know about H. pylori. We know about different types of antibiotics, how they work. So H. pylori, it has a gram-negative cell wall. So let's give an antibiotic to break apart the cell wall, which is similar to penicillin. But instead of penicillin, we give amoxicillin and also the inhibitor drug, so it doesn't get broken down. So in total, we give them something called a co-amoxiclav as one of the 3 drugs we give them. So we give them a co-amoxiclav to break down the cell wall. If that fails, we can give other mechanisms of antibiotics. And I said there was one mechanism of antibiotics that works by blocking off the ribosomes, the 70S ribosomes. And if we block the 70S ribosomes, that bacteria can't synthesize proteins, it dies. And that antibiotic is called clarithromycin. It binds onto 50S ribosomes, prevents protein synthesis. I also told you that people with H. pylori, they suffer with excess acid because what bacteria has done is neutralized it. So by negative feedback, because our kind of stomach knows that there's not enough acid to digest food, it produces too much of it. And that causes some symptoms that are really uncomfortable, so heartburn, those kind of things. So we also give them another drug to kind of stop the production of acid. So they're called proton pump inhibitors. These are blocking of cells that produce acid. And there's a drug it's used called omeprazole. Omeprazole prevents hydrochloric acid being released into the stomach, which relieves the symptoms for people. It doesn't kind of completely block it off, it only partially does that. Again, that's to do with the dosage that you give. So what you give with someone for H. pylori, this is kind of standard practice now, is you give them something called triple therapy. So 3 mechanisms of different drugs to kind of eliminate it by all means. So we give them a cell wall inhibitor to break down the cell wall. If that fails, we give them a ribosome to prevent protein synthesis, a ribosome inhibitor. If that fails, to kind of relieve symptoms, we also give them a proton pump inhibitor to release, to slow down HCl production in the stomach. So we're relieving symptoms at the same time killing off the bacteria. That's called triple therapy. However, if left untreated, as I said before, it can progress into more severe consequences if left untreated. This is really important as future clinicians, as future healthcare professionals, to know what's going on and diagnose it early. So if left untreated, it can cause things called peptic ulcers. So basically ulcers in the surface of your stomach, which can start to bleed as well if left untreated. And if that bleeding occurs, you can bleed internally, lose a lot of blood. And obviously if you lose a lot of blood, that can lead to death. So these are really serious stuff. You can also get something called gastroesophageal reflux disease, which is too much acid going in your esophagus. Kind of acid in your esophagus starts to break down the lining of your esophagus. And that leads to something called Barrett's esophagus. So you can see over here the epithelium of your esophagus is getting broken down by acid being present there. It's normally not present there. If that continues to happen and left untreated even further, you can lead on to gastric cancers, so cancers of the stomach, cancers of the esophagus, those kind of things. So it's really serious stuff. So last thing, let's kind of link everything together. So I told you one symptom was blood in your stools. Blood in your stools comes from these peptic ulcers bleeding out. You kind of digest your blood, you get blood in your stools. The reflux disease, the uncomfortable heartburn, comes from acid going up your esophagus. Weight loss because the bacteria are using up your food. And also you're bleeding, so you're kind of losing nutrients as well. There's a lot of things, and you guys know now how to link all these 3 things together. So what we've done today is we've talked about the mechanisms of, first of all, how to classify all bacterial species. We've talked about the gram staining process, which is kind of really important nowadays. We've looked at how to kind of further specify what bacteria the person's suffering with, so kind of different shapes, different symptoms, those kind of things to narrow it down. We then talked about a really common patient case, which is H. pylori. So H. pylori, I told you, infects about 50% of the UK's population, so really common bacteria, but only in certain people it can aggravate and cause symptoms, as we've discussed. We talked about the symptoms they'll present with. We talked about how H. pylori is going to cause its effects. We then talked about the whole mechanism of how general antibiotics work. We then talked about the specific ones of penicillin, amoxicillin, and how I linked it towards enzyme inhibition, so non-competitive and competitive. And then talked about how these drugs actually work. And then talked about how to test and diagnose for this condition called H. pylori, caused by the Helicobacter pylori bacteria. We talked about the principles of the Urea breath test and also ELISA test and monoclonal antibodies, which again is really relevant for A-level biology. And then we summarized talking about treatment and if left untreated, the long-term impacts. So thank you very much for listening. If you guys have any more questions, leave them in the chat. We'll go through them now. But if you liked my style of teaching, you want to see kind of more of this, or you're interested in getting tutoring for A-level biology, let's work together to get the grades you deserve. I know a lot of you are currently working towards your application in October, and I know a lot of you also have mocks coming up in September, which are really crucial to getting into the university of your choice. Let's work together and get those grades. So I offer the mentorship scheme that I've advertised before. So I'm offering now chemistry and biology. And you can see what's listed over here. So obviously DME, we can kind of discuss the next steps. I offer one-to-one services and also group services, which obviously will cost less, be more cost effective for you. for one-to-one if you need extra support. I'm also offering one-to-one support. We also go through your personal statement, your application to university. You have full control over what topics are taught, what notes you would like, what PowerPoints, those kind of things. So definitely get in touch if you're interested. Okay. So thank you very much for listening. If you have any more questions, leave them in the chat. My Instagram handle, also my email address is over there. So definitely give me a follow and you'll be alerted of any kind of future webinars or other kind of content I'll be uploading. But again, thank you very much for listening. If you have any more questions, leave them in the chat about anything you want to ask me, and I'll stick around for a couple of minutes and answer them. So why is bloating one of the symptoms? So remember I said Helicobacter pylori releases urease, the enzyme that breaks down urea into carbon dioxide and ammonia. We talked about how ammonia neutralizes the stomach acid. And then carbon dioxide is a gas. Gas is going to cause bloating. That's why bloating leads to excessive birthing. So it's all really linked together. I hope that answers your question. So if you guys have any other questions, obviously leave them in the chat about anything. If not, thank you very much for attending. This PowerPoint and the recording of this lecture will be on YouTube soon. Again, thank you very much and enjoy the rest of the day.